Hey guys, happy Friday. So we are here talking about our last piece of chapter 17, which is the translation process of taking that RNA molecule made during transcription and then turning it into a chain of polypeptides or a chain of those amino acids that makes up a polypeptide. So genetic information flows from the messenger RNA into protein through the process called translation. Molecular components of translation. So a cell's responsibility is to translate the mRNA message into a protein with the help of transfer or tRNA. This was on your worksheet. So basically, transfer RNA is the opposite of the RNA molecule. So if you're looking at a 3-code sequence of CCC on messenger RNA, the transfer RNA would be the mirror image of that being GGG. Transfer RNA's job is to carry the amino acid from the cytoplasm into the ribosome to help complete the growing polypeptide within that ribosome. The translation process is complex in terms of its biochemistry and the mechanics behind it. So overall, this is everything that goes on with translation at once. So this pinky red is your ribosome. And then you have 3 different sites inside your ribosome. You have 1 site, 2 site, and 3 site. So this is your growing polypeptide. So each amino acid being added in. These blue cylindrical structures are your transfer RNA carrying that message to pick up the polypeptide. They bring it in, it binds, and then they let go. Well, I guess we're going in this direction. So comes in, it binds, it lets go. The structure and function of transfer RNA. The molecules of transfer RNA are not identical. Each carries a specific amino acid on one end, and the other end is the anticodon, which is complementary. It's the opposite base pair of that 3 nucleotide codon in messenger RNA. A transfer RNA molecule consists of a single RNA strand that is only about 80 nucleotides long. It gets flattened into the plane to reveal the base pairing, and the transfer molecule can then look like a clover leaf afterwards. So chemically, it's that 3-leaf clover. 3-dimensionally, obviously, it's much more complex. And then we use this cylindrical symbol in the book. So down here is your anticodon, and then your amino acid attachment sites up here. Because of hydrogen bonding, transfer RNA actually twists and folds into a 3-dimensional shape, and it creates something that's roughly L-shaped. So there are 2 different steps that lead to accurate translation. First is the correct match between the transfer RNA and an amino acid. So first you have to get the correct amino acid to the transfer RNA anticodon. Second is a match between the transfer RNA anticodon and the codon in messenger RNA. Flexible pairing at the 3rd bases of codons is called a wobble, and it allows for some transfer RNAs to bind to more than one codon. This goes back to that codon chart I showed you last time and the one I posted in Classroom, that there are many amino acids that get coded for by more than one codon. So arginine is one, it's coded by 6 different codons. That's the wobble. So in those instances, the first 2 are almost always the same. That 3rd one allows for some fluctuation. So here, this is how we get our amino acid to attach to our tRNA. It's this very complex enzyme. The amino acid goes in, it uses ATP or chemical energy to bind, the transfer RNA comes in, and then it lets go. Ribosomes are responsible for facilitating the specific coupling of transfer RNA anticodons with the messenger RNA codon during protein synthesis. There are two ribosomal subunits. One is large, one is small, and they are named accordingly. And they're made of proteins using ribosomal RNA. Bacterial and eukaryotic ribosomes are somewhat similar, but they have differences in their structure. Some antibiotic drugs in bacterial cells specifically target the bacterial ribosomes that we are trying to attack in our body without harming the eukaryotic ribosomes. We are going to focus mostly on eukaryotic translation because prokaryotic gets very confusing. So ribosomes have 3 binding sites for the transfer RNA. The P site is responsible for holding the transfer RNA as it carries the growing polypeptide chain. The A site holds the transfer RNA that is carrying the next amino acid that needs to be added. And then the E site is the exit site where it gets rid of the transfer RNA molecule. In building a polypeptide, there are 3 stages of translation. These are going to be very similar to the stages of transcription: initiation, elongation, and termination. All 3 stages require protein factors that lead the translation process. Ribosomal association and initiation. So the initiation stage of translation brings together the messenger RNA, a transfer RNA with the first amino acid, and the 2 ribosomal subunits. First, the smaller ribosomal subunit binds with messenger RNA and a special indicator, an initiator, transfer RNA. Then, the small subunit moves along the messenger RNA until it reaches the start codon. The start codon is always AUG and codes for methionine. Always AUG, that is always the start site. Proteins called initiation factors bring in the large subunit that completes the translation initiation complex. So here we bind in, it finds that AUG, the anticodon UAC, it grabs a methionine. There's multiple different ways to say that, but I usually say methionine, and then it can start the process. Elongation. During elongation, amino acids are added one by one to the preceding amino acid at the C terminus for the growing change. Each addition involves proteins called elongation factors that occur in 3 separate stages. First is codon recognition, second is peptide bond formation, and third is translocation. Translation proceeds along the messenger RNA, or the mRNA, in the 5' to 3' direction. So start to finish, here's your P site with your growing polypeptide chain. Your A site comes in, it grabs on, let's go. So it passes it on. This tRNA is going to move to the exit, and this tRNA is going to move into the P site while this one leaves. And that keeps going round and round and round until we are finally done our polypeptide. Termination occurs when a stop codon in messenger RNA reaches the A site of the ribosome. The A site accepts the protein called a release factor. The factor causes the addition of a water molecule instead of an amino acid, and this reaction will release the polypeptide from the translation process, and then it comes apart and can be transferred to the part of the cell where it is needed. So we're not going to really worry about single-celled organisms, but looking at eukaryotic cells, so we've come about a stop codon here. It adds the release factor, which adds a water that stays in the A site. We lose our tRNA, and our free polypeptide is free to go. The ribosomal large and smaller subunit will break apart from one another to let go of this RNA. This RNA can actually now be reused. So if we need more of this polypeptide, this RNA can re-enter back at the start site of our ribosome and be read through once more. All right, we are going to stop here for today. Thanks, guys, for me this week. And as always, if you have questions, just let me know. Don't forget your worksheet is due tonight at midnight. That worksheet that was assigned on Wednesday.